Hey everyone! Today we're diving into something that sounds like pure science fiction, but is 100% real science. I'm talking about growing living cancer cells in a lab on purpose to figure out how to destroy them or even stop their growth. It's called cell culturing, and it might just be one of the most important tools we have in the fight against cancer. So, what even is cell culturing? Here's the basic idea. Our body is made up of trillions of cells. Yeah, you heard me, trillions. Scientists take samples that contain some of those cells and they grow them outside of a living body, in a dish, in a lab. Now I know what you're thinking, how? For a cell to grow, doesn't it need to be in our body? Well, not necessarily. We can replicate our body's environment in a dish. All we have to do is make sure that the temperature is just right, the nutrition is available, and also that the pH is just right. Once we have all three of those things, look at our cells. They're growing. Think of it this way. It's kind of like we're setting up the perfect little apartment for a microscopic tenant. I mean, come on, if I were a cell and I had to grow here, I don't think I would have any complaints. It seems perfect. I have everything I need. Now, here's why cell culturing matters so much in cancer research. Before doctors can give a patient a new drug, they first will want to know, does it work? And more importantly, is it safe for the patient? Testing new drugs on humans first is way too risky. So what happens is cell culturing, of course. Scientists will grow cancer cells in their culture dishes and apply drug treatments directly to those cells. After some time has passed where our cells and our drug have gotten acquainted, scientists are going to make observations and ask questions. Did cancer cells die? Did cancer cells simply stop growing? Did they shrink? And more importantly, what effect is this drug going to have on healthy cells? Once the drug has passed all the tests in the culture phase, what usually happens is it moves on to a human clinical trial. During this time, the drug will be tested on many different patients. If the desired results are achieved, the drug can eventually be sent to the FDA, which is the Food and Drug Administration. They're the ones that will give the final approval for the drug to be used in practice. Here's the fascinating part. Not all cancer cells behave the same way. A skin cancer cell is different from a cancer cell that originates in the lung, so the treatment will more than likely also be different. Each tumor cell could have similar environments, but their differences are what cause the treatment methods to be different. To go even deeper, let's examine the subtypes of breast cancer. I know what you're thinking. Breast cancer is breast cancer. There's only one kind, right? Wrong. Here we have 4 examples, but we're only going to focus on the 2 circled in red. So, what makes these 2 different types of breast cancer different? Well, I'll tell you. On each cancer cell, we have different types of receptors. Now, the triple-negative breast cancer, as you can see, has a lot of negatives. Focus on that HER2 receptor. The inflammatory breast cancer is quite the opposite. It has a lot of positives, meaning all those receptors are present. In our treatment, the receptors are important because the receptor is what the drug is going to bind to on the outside of the cell. So for example, if our drug targets HER2 positive cells, that means that unfortunately for patient number 1, this drug will not be compatible because they're HER2 negative. However, patient number 2 may be a good candidate because they're HER2 positive. This is why cell culturing is so important for cancer research. It allows scientists to test different drugs on different types of cells. So next time you hear about a breakthrough cancer drug, remember, it almost certainly spent a lot of time in a culture dish first. Cell culturing may seem basic, but it's a huge impact on science and medicine. It's quiet, careful, and meticulous work, but it saves lives. Who knew something so small could make a big impact? Thanks for watching, and remember, stay curious.